So I will tell you about what is genetic epidemiology. That's most of it, and big data I will refer to as the lifelines cohort, which Bandana also already mentioned a bit about. Ooh, that's going quick. So indeed, genetic epidemiology, lifelines cohort, and then as hypertension is one of an example that I'll give on the research that we are doing. So epidemiology, you may all be familiar with that. In epidemiology, we are studying health and diseases in large populations. And we are looking into what lifestyle factors or other factors are causing that somebody gets the disease and others stay healthy. And also these factors can interact with each other. For instance, someone who has very healthy lifestyle is eating not very calorie rich food, is doing exercise, but he may get, well, maybe a virus, or due to pesticides, he can get a disease, even though he is living a very healthy life. And of course, in genetic epidemiology, we are looking how genetics play a role in this, in health and diseases of the population. And also these genetic factors can interact with lifestyle and with external factors. So, but before I will talk more about epidemiology, genetic epidemiology, I would like to introduce you a little bit more into the lifelines cohort, which is a big data cohort that that is collected in the northern of the Netherlands. It's a 3-generational cohort and aiming to study population health and eventually precision medicine in the northern of the Netherlands. Oops, it's going very fast. So in this in lifelines, data had been collected about more than 165,000 participants. 145,000 of those are adults. And the aim is to follow up these individuals more than 30 years. So now we are almost at 20 years, so over halfway of the study. And as I said, it's typical for the lifelines cohort is that it's a 3-generational design. So initially, the individuals in the age range of 25 to 50 were asked by their GP to whether they would like to participate in lifelines cohort. And then these people were asked also their children to participate in lifelines and their parents. So it's really a unique thing of lifelines that we have these generations. And by having these generations, you can look into how diseases are transmitted from the grandparents to their middle generation to the children and grandchildren. So initially, we more than 300,000 individuals were invited through their GP, and 25%, so more than 80,000, were positive about participating in lifelines. And these 80,000, they asked their family members about these family members. About 65,000 were added because they were family members of this middle generation. And also 5%, you could also self-register via a website. That's about 20,000. So in total, we have 67,000 participants in lifelines starting at baseline. Then this lifeline study started in 2006, and the first, well, of course, the startup was a bit slow. So the first 167,000 individuals were collected from 2006 to 2013. Then, you see here, and I think I've done the next slide. Yeah. So we're now at 2026, and we are currently, Lifelines is collecting data for a 4th wave. And, well, of course, as you can imagine, partition rates dropped. In the third assessment that was started in 2020 till 2023, only about half of the 50 to 60% of the lifelines of these 167,000 individuals only participated in the third wave anymore. So in the fourth wave, Lifeline started to expand the cohort by including new participants. Then, at each wave, so that was every 5 years, individuals had to come to a Lifeline's location to do physical measurements like height, weight, body pressure, blood pressure, ECG, lung function, cognition, and psychiatric interview. But also every 1.5 years, and also at these 5 years, they had to fill in a lot of questionnaires, but also within these 5 years, there was about 1 to 2 questionnaires that they had to fill in with additional data on medical history, lifestyle, quality of life, mental health, social network, daily activity, and sleep. And also dizziness was with 2 questions in this questionnaire. So one, Bandana said all about it. Dizziness with falling within the past 12 months. And another, how much did you suffer from dizziness in the last 7 days? And of course, we also have genetic data. At every 5 years, blood was collected. So from these 167,000 individuals, the first set of genotype data was released in 2016. That was about 15,000 samples. Then in 2020, it was extended via the UGLY consortium, which is the UMCG Genetics in Lifelines initiative. So the first part was actually paid by Lifelines, but then we wanted to expand it. So the UGLY consortium was founded with where money was collected from all PIs from within the UMCG. And then there was enough money to have 36,000 more individuals with genetic data. And this was expanded even in March 2023 with another 28,000. And recently, last December, it was even extended with more 32,000 more individuals. So in total, we now have one genome-wide genetic data set with more than 110,000 participants. So still 57,000 are not typed, and there are some initiatives to type some of these more patient-specific cohorts, but there are still no genotyping efforts being done to type these, but ultimately we would like to have all 167,000 Lifelines participants with genetic data. So, but from all these you can see that Lifelines is a rich data set for epidemiological research, and of course we focus on genetic epidemiological research. Not working. So remember this, that genetic epidemiology was about health and diseases in large populations. So mostly we work on Lifelines. We will also have some other smaller cohorts, but mostly Lifelines. And to see how genetic factors are involved in health and disease of the population, and how it's influencing with lifestyle and other external factors. So in genetic epidemiology, we mostly have 3 main research questions. So the first one, what we like to know, is the disease genetic? If it's not genetic, then it's no use to do any further statistical analyses. Then the next question is, of course, when we determine that a trait is genetic, we would like to know which genes are involved. And then the third is, can genetics be used in prediction? So is what we found out which genes are involved. We would like to know, can we tell when a patient comes to the clinic, can we tell, well, you have a higher chance to have a more severe or more quicker progression of the disease. And of course, we would also like to do some, ultimately some screening so that you can use genetics to prevent people to get diseases. That's still very well in the distant future. So the first question that was about, is the disease or trait genetic? These are so-called heritability studies. And ideal, you all probably know that this phrase, the disease runs in the family. So if the grandparent has a disease, and his daughter also has the disease, and two of her children also have the disease, then you could start thinking, okay, might there be something genetics going on? So this is called familial aggregation. So it aggregates in the family. The disease aggregates in the family. But is it genetics or is it shared environment? We don't know. At this point, it could also be that, well, it's the lifestyle of and the shared and so on, the shared environment that is causing that this individual, that the children also become sick. So in heritability studies, we want to determine how much of individual differences in a trait or a disease, how much is it due to genetic factors, how much can be explained by familial or shared environment, and how much is unique to an individual. And the perfect study design to do this is twins. Because with twins, you can distinguish shared genes from shared environments. And that's because you have twins that share all of their DNA, the monozygotic twins, and you have twins that share half of their DNA, the dizygotic twins. But both dizygotic and monozygotic twins, they are, if they're raised in the same family and not adopted, then they will also share they will both fully share the shared environment. So that's in this way you can perfectly discriminate between what's shared environment and what's genetic. And then if you would look into the resemblance of a trait, in this case it was its reading ability, then you will, if a trait is genetic, then you will see a higher correlation among monozygotic twins than among dizygotic twins. And if you see this, so a higher phenotypic correlation in monozygotic twins, then you have evidence for genetic component. Okay. So what the heritability analysis is about is looking into these correlations or covariances. And that's actually, this is a twin, but you can also extend it to families. But then in families, it's difficult to discriminate between the shared environment and the shared genetics. So then we're mostly talking about a familial component instead of a genetic component. Okay, so then I said, once we have established that there is a genetic component, we want to know, okay, then what genes are involved? And as a genetic epidemiologist, we are focusing on common diseases, which are diseases that occur frequently in the populations, at least with prevalences over 1%. And then most of the definition of these common diseases, it's that it's complex diseases. These diseases are mostly caused by multiple genes that are interacting with each other. They're interacting with environmental, lifestyle, other factors. So it's really complex diseases that we are interested in. And because we are looking into populations, so we shift from inheritability studies, we look into in families, in population, to look in for common diseases, we have to look into populations. And for populations, we look into association studies. And well, I think about 20 years ago now, the technology made it available to do genome-wide association study, which means that we can analyze millions of genetic variants across the whole genome, so on all of the chromosomes that you have in your body, to find genes or that are involved in common diseases or traits. And well, this is called a Manhattan plot. You may guess why, because you see all these high peaks. And what these peaks mean is that, well, what you see here on the x-axis is that you have chromosomes. So that's the whole genome from chromosome 1 to chromosome 22. And each dot represents a genetic variant that has been analyzed. And the significance is plotted here on the minus log 10 scale, meaning that the higher the points, the higher the significance, so the more evidence that this gene or that this genetic variance is associated with a disease or trait of interest. So I don't know how much you know about genetics, so I have a couple of slides on how genetic variants, what do we mean with genetic variants. Well, of course, you have the DNA, which is in the cell nucleus, and these of the chromosomes, and these are built up on DNA. And the DNA is wrapped around histones, and when you unfold it, you get this DNA double helix. And these double helixes, these are built up of actually only 4 different base pairs. So it's the cytosine, adenine, guanine, and the tyrosine. So for each of these 22 chromosomes, you get 1 pair from your father and 1 pair from your mother, and you have these chromosomes pairs. These are built up of these AC, G, and T base pairs. So everybody, well, along the chromosome, this is the whole DNA sequence is built up of these AC, G, and Ts. When you look at 2 different chromosomes, then there will be, well, 99.99% identical, but at some places there is variation. For instance, the maternal chromosome has an A at this position, so an adenine, and the paternal chromosome has a cytosine at this position. So this is a single nucleotide polymorphism, or a SNP in short. So it's just a single base pair substitution. So some people have an A in this position, other people will have a C in this position. And that's what we measure. So along the chromosome, okay, some further terminology. These A and C, these are called alleles. So that's the different variations that's possible on these for this particular SNP. And well, because you got this from your mother, this from your father, the combination of these 2 alleles, AC, it's called your genotype at this position. So what is being done in a genome-wide association study is that we have chips. We have these microarrays on which there is there are probes for, well, more than 500,000 of these base pair variations, these SNPs. And we have the DNA, well, in this case, let's say from patients and from controls. And all of these DNAs, is put on these chips. So also on, well, for every patient, one chip. And these data is then processed. And then for each person, you get the genotypes of all these 500,000 SNPs. And you can analyze them statistically. So if you analyze each of these variants separately, and if you, well, in this case, a disease, patients versus controls, so the analysis is actually quite simple. It would just be a logistic regression analysis. And then you plot the significance, these p-values, on this so-called Manhattan plot. The theory behind this genome-wide association studies is the common disease, common variance theory. So it means that we focus also on common variants that occur at least with a frequency of at least 1% in the population. So these chips have been designed in such a way that, well, there was this human genome project. I don't know if you heard about it in the early 2000s, that all the humans' DNA was sequenced, and then from these sequence data, this human genome project derived, okay, the variations are here and there in the genome, and these variations were they picked 500,000 of these variations to put on the chip. This also means that with genome-wide association study, we barely have power to detect variants that have a frequency less than 1%. So it's really the common variants that we are investigating with genome-wide association studies. Because we are looking into 500,000 or more SNPs, there is this huge multiple testing burden because we test each of these variants individually. So we do 500,000 tests, and we cannot say simply with the nominal significance threshold of 0.05, you would see many false positive results. So we have to really be strict on the genome-wide significance threshold, which means that we also need large sample sizes to detect variants. So when we first started with genome-wide association studies in the mid-2000s, 2005 or 2006, the first genome-wide association study was published. Actually, they were pretty, we were disappointed with the results because we thought, okay, we do the genome-wide association study. Once we do that, we will know everything about every disease. But unfortunately, for instance, if you look into height, each of these dots represents a genome-wide association study for that trait. So the first studies for height, for body length, no associations, no genome-wide significant associations were found, even with sample size. So we started off with a sample size of 3,000. Well, it appeared to be too low. Then we increased the sample size up to 10,000. Still no success for body height. While we know that body height is heritable for 80%. 80% of how tall you are is defined by genetics. So why don't we find anything? Only when we started increasing the sample size by so combining data from multiple cohorts, we eventually started finding genetic variants that are associated with height. And currently, so this is from a paper in 2012. So currently with height, we analyze data combined data from more than 1 million individuals, and I think we are up to 2,000 variants that have been associated with height. But so by forming consortia, we combine data from many cohorts and meta-analyze the results from each of these cohorts. More and more SNPs were found. And this is a second paper on 10 years of GWAS in 2017. And we see for various numbers of traits that the number of SNPs, number of genetic variants that have been associated with diseases increased and increased. So what did we learn from genome-wide association studies? First, of course, we know that we found genes that are associated with disease. We found genes which are logical candidates, which we knew before, but also we learned, we found much more genes that, well, we didn't think of as being involved in the disease or trait, but they were. So with these genes, with all these genome association studies, we started to learn more and more how diseases, about the disease etiology. Also, sometimes we could identify drug targets with genome association studies. That's not really our field. So, but pharmacogenetics is also really interested in genome association studies. And also what we learned is, well, as I said, we thought with genome association studies that once we have performed that, we would know everything about the disease, but it appeared to be much more complex than expected. And the genetic variants that we found only had very small effects. So can these variants, can we use it still for risk prediction? We thought, okay, when these, so we started to think, how can we use? So we find for height, as I said, we have now 2,000 variants that are associated with height. How can we use this for risk prediction? How can we tell if one person has, well, higher risk for being, well, being taller, but of course you can also for getting a higher blood pressure, or to get, well, with bandana we are going to do dizziness. Why would somebody get more dizzy? Which genes would be involved in that? So the next thing is, so we started to look into risk scores by combining all these genetic variants that had small effects. So this is from a paper from the earliest GWAS. The first GWAS is into blood pressure, so systolic blood pressure on the left, this diastolic on the right. And what you see, these were the first, well, 6 and 9 variants for these two traits. And what you see on the y-axis is how much variance do they explain. And this is really variance. It's not proportion. So it's less than 1% in variation of blood pressure that was explained by, well, the most significant genetic variant that we found. So these effects are really, really small that we find. So we started thinking, okay, we do find 6 hits for systolic, 9 hits for diastolic blood pressure. Can we combine them in a so-called polygenic risk score? So we started, well, off with just a simple score, combining all these data. So we have a number of SNPs. So let's say it was N. This N was 6 for systolic blood pressure, and we just do a weighted sum where this D is, well, we have 6 SNPs that we have for systolic blood pressure, we measure these 6 SNPs in the population. So let's say in lifelines, we know we can determine for these 6 SNPs how many risk alleles does an individual have for systolic blood pressure. So this could be either 0, 1, or 2. So you have genotype, so you have either inherited none of the risk alleles from your father and mother, or 1 or both. So this is just a score of 1, 0, 1, or 2. And then the next thing is what to do with these weights. We can just have a weight of 1, where we just count the number of risk alleles per individuals. That's a possibility. But we could also use genome-wide data, association data for it. So we know. So if we go back to this plot on the these proportion of variances, you see that not all variants have the same effect on the outcome. So you can give more weight to SNPs that have a stronger effect so have stronger association with the outcome. So from genome-wide association studies, you can get such an estimate for the effect, and we use that as a weight in this formula. So for instance, here, if this is with 3 SNPs, so 3 variants, and these have been identified as the risk alleles, let's say for blood pressure, and these were the effect sizes. So the allele A increases blood pressure with 0.013 millimeter mercury. Then if an individual is homozygous, so it has 2 A alleles, meaning that then the contribution to the polygenic risk score would be 2, because of 2 A alleles times the effect size times the beta. And that you do for all of the SNPs. So there's 1 C over here. So you count this 0.045 one time. Here, this is this individual is TT. So there are no risk alleles. G was the risk allele. So this would be 0 times the effect times the beta. So in this way, you end up with a polygenic risk score. It's just a number, ranging in this case from 0, if these would all be not carriers of the risk allele, up to, well, 2 times the sum of the betas. So it's just a continuous score that you get, and that will be different for all individuals. And of course, if you include more SNPs, then you will get more variation in this polygenic risk score. So we started off with polygenic risk scores with just the variants that were found genome-wide significance. And this is 5 times 10 to the minus 8. That's the value, significance threshold for genome-wide significance. And I say here independent because there's correlation structure in the genome. So 1 SNP and 2 SNPs that are next to each other, they are likely very highly correlated to each other. So they will not contribute independently to the association to the disease. And then then we didn't explain much of the heritability when we used this score. So people start, okay, maybe we don't find, or we are too strict with this 5 times 10 to the minus 8. We lower the threshold, and that seems to improve the polygenic risk scores. And then methods were developed where we include all SNPs, regardless of the significance threshold, because you do weigh them by their effect size. The SNPs that are non-significant, or then they will only have a small effect. And you correct for the correlation structure within the genome. And the most recent advancement is to also include functional annotation. So that's SNPs that have an effect on the protein, like missense variations or variants that influence the expression of a gene. They get higher weight in the, so then this W will be increased compared to intronic SNPs or SNPs that are even in between genes. So for each of these individuals, you just get one score S. And you can treat this score S as any other variable in your model. And what we then, to determine what's the contribution of genetic risk to the disease, we usually have two models. One where you have just the covariates and determine, okay, so what's, let's say, like age and sex. So how much does age and sex explain in my trait of interest? And then you add the polygenic risk score, and then you get another number on how much on the the percentage of variance explains, and you look into this difference. And that will be the percentage of variance that can be explained by the polygenic risk or by the genetics of this trait. Okay, anything clear so far? So then let's go to our example. So as I said, in our unit, we're mostly focusing on cardiovascular disease, and blood pressure is one of our highlights. In hypertension, well, hypertension is occurring a lot, prevalence of over 30% in adults, and heritability of blood pressure is around 50%. So half of the variation in blood pressure is determined by genetic factors. What we did, well, already since the early 2000s or mid 2000s, we started looking into blood pressure. And at first we started looking into hypertension. So do you have high blood pressure, yes or no? But then we didn't find that much. Then we started, okay, maybe blood pressure itself is better to look at. And that was because then you have a continuous scale instead of this dichotomous scale. So GWAS, Genome-wide Association studies looking into hypertension are mostly focused on this continuous blood pressure variables. There have been a lot of efforts. So there has been a consortium, the International Consortium for Blood Pressure, who meta-analyzed data from 77 studies, among which Lifelines. And well, that was published, but there were also other consortium, other cohorts or consortium also working on blood pressure. And I think in around 2020, we said, okay, let's combine our efforts to have a data set of more than 1 million individuals and see what's coming up. Yeah, for what do we found for 7.5 million common variants for systolic blood pressure, diastolic blood pressure, and pulse pressure. And this was published in 2024, 2 years ago, and we found, in total, well, these are the 3 different GWASs for systolic, diastolic, and pulse pressure. And we found in total about more than 2,000 variants that are influencing blood pressure in one way or another. Of these, variants, about 100 were novel, so they were not found in these individual GWASs, but just by combining, we found them. And as you can expect, these effects were smaller than in the individual cohorts. So with more and more data, combining more and more samples, you can find more and more genetic variants, but with more, even larger, smaller and smaller effect sizes. So what about, so we have these 2,000 variants. What if we combine them into a polygenic risk score? Well, for instance, for systolic blood pressure, all these 2,000 variants together, they now can explain 11% of the variance in blood pressure. So remember that the heritability, so of blood pressure, so was 50%. So 50% would be the top, the max that could be determined by genetic factors. So we're there at 11.37%. You would still think, okay, this is not that much, only a quarter. But remember also that I said with GWAS analysis, we are focusing on the common variants. With GWAS data, we can also determine what would be the maximum number of heritability that can be explained by common SNPs, and that is 17%. So meaning that 17% is the max that we can reach with genome-wide association studies. The rest, so the remaining, what is it, 33%, that's coming from the rare variants that we do not capture with genome-wide association studies. But if you look into this difference, 11% versus 17%, then you see that we are there almost reaching up to 2/3 of the variants that we have found yet with genome-wide association studies. And if you look into, to visualize the effects of the polygenic risk scores, what we often do is then cut the polygenic risk scores into centiles, or deciles, so into 10 categories, and we can compare. We can, in each of these bins, each of these deciles, we determine what's their mean blood pressure, and then we can see from this that there is about a 17 millimeter mercury difference between the lowest and the highest category of the polygenic risk score. So that's a pretty big difference, and 10 millimeter mercury for diastolic blood pressure. If you look into hypertension on the right, we can do the same. And here then the odds ratio is plotted. So the risk, how much more risk do you have when you're in the top decile, so have the most genetic variants, compared to the lowest decile, having the least genetic variants for blood pressure. Then you see that individuals in the highest category have more than 7 times increased risk of hypertension compared to the lowest quartile. So these are numbers, this 7. That's you could easily these could be used in a clinic, because genetics, it's already fixed at birth. So when you would determine what variants do you have at a young age, then you could, for instance, you can determine which individuals have a high risk for getting higher blood pressure. You could maybe follow them more frequently, or give them earlier advice on a healthier lifestyle. So indeed, polygenic risk scores, I think there's a future to be used in precision of personalized medicine and prevention. So to identify those at a high genetic risk, and advise them to adapt their lifestyle to prevent disease or to slow down the progress of the disease. Could also be used for treatment advice. Also, pharmacogenetics could be a goal or an option for polygenic risk scores. Okay, to summarize, in genetic epidemiology, we focused on three questions: heritability analysis to determine if a disease is heritable, then genome-wide association studies to identify variants and genes involved, and we learned over the years, well, it started off slowly, but now we have identified, with GWAS, identified many variants for many diseases. Polygenic risk scores can be used in personalized medicine, as shown in this example. With that, I'd like to thank the organizers for inviting me here, and my boss, Professor Harold Sneeder, and colleagues from my department and Lifelines for their data, and of course you for listening. Thanks.